Well, Conway's lab says yes, so it must be good. Oh, we're on the hour. Thank you all for showing up at the peak of vacation season. It is my sincere pleasure to humbly introduce me. Some of you in the audience don't know who I am. My name is Ed Pryzdial. This is how Mama taught me to spell it. This is how Mama taught me to say it, Pryzdial. I did my PhD in a plasma protein pathway called complement, part of the innate immune system at the University of Toronto. Then I jumped ship, not too far, still in a plasma protein pathway, coagulation. I went to the United States at the University of Vermont, the National Institute of Health Center of Excellence to learn about coagulation. And ultimately, I found myself here after doing a professorship at the University of Ottawa for about a decade. Now I'm clinical professor in the Department of Pathology and Lab Medicine, and I am a steadfast supporter of the Center for Blood Research since prior to inception, when the Center for Blood Research was still just a little sketch on the blackboard. So I've been around for a little while here. Well before this building was even a hole in the ground. I am mostly, my primary appointment is as a senior scientist with Canadian Blood Services. I am very pleased to introduce 4 people who will be speaking today: a postdoctoral fellow, Li Hua Hao, and 4 PhD students, Dr. 2B status, Alexandra Witt, Henry West, and Jean Perrier. And what we do in my laboratory is coagulation biochemistry. We focus on various aspects of coagulation, and you all know that clotting happens because of coagulation. Anybody not know anything about coagulation here? 0? There's people in my laboratory putting their hand up. That's kind of okay. We're going to be talking in my lab. Well, we do in my laboratory clot busting. We also talk about, we think a lot about how viruses interact with the coagulation system. And we also think about how clotting deficiencies work. Today, we're going to tell you a little bit about how work in our laboratory involves clot busting or fibrinolysis. This is the physiological pathway, fibrinolysis. When clots occur where they're not supposed to, it's called thrombolysis when we use a therapeutic to bust up the clot. And we're going to talk a lot about that today. We're also going to tell you about how viruses hijack the coagulation system to cause pathology and to enhance their own infectivity. We're not going to talk much about the molecular basis of coagulation deficiency at all today. Oops. Now, factor 10, or factor X Roman numeral, is at the center of the coagulation universe, as depicted here in this schematic. Take-home messages, well, really highlighting that it's at the center. We're not going to talk too much today about this, so I'll just get rid of it, even though some people in this audience I know work on that part of the pathway. But you can really see that there's a branch in the coagulation pathway that culminates at the activation of coagulation factor 10 to factor 10A. Anytime you see an A in a clotting factor name, it means activated. In this pathway, I've depicted spheres as functional proteases and ovals as their cofactors. And you may see that a lot of these cofactor enzyme complexes assemble on the surface of a phospholipid membrane. And those phospholipid membranes, they have to have anionic phospholipids, such as phosphatidylserine, for these proteases to associate. So that's an important theme in coagulation. Phospholipid membrane has to assemble the cofactor and enzyme complex to get the proper acceleration that is required to amplify over intrinsic inhibitory pathways that circulate around in your plasma and blood. Factor 10A being at the junction of both the extrinsic initiating pathway and intrinsic amplifying pathway, then goes down in association with its cofactor on a membrane to produce the biological muscle of coagulation called thrombin. That makes the clot. But thrombin, as well as other proteases in coagulation, have this thing that most of you in this in this audience don't know about, and that is they signal cells. And that gives coagulation the potential to be part of just about every sickness that you can think of, everything from diabetes to cancer. But also, they're very, very important constituents in many normal biological processes, like development, angiogenesis, as an example, the development of blood vessels. So we're going to tell you today, first, Li Hua is going to get up on the stage, and she's going to tell you about how these different enzyme complexes, called tenases, because they activate factor 10, how they differ in how they recognize factor 10. She's going to compare these two, which exist in mammals. She's also going to compare it to one that is non-mammalian, non-human, I've written here. She's going to look at one that comes from snake venom. What we're going to do is we're going to tell you about how factor 10, a clotting factor, gets modulated and gets converted into a clot busting agent. And we're trying to harness that potential into a new thrombolytic drug, a clot busting drug. And Alexandra is going to tell you about that. And that work is in collaboration with Li Hua. Following that, we're going to jump up here and talk about this tenase, this tissue factor, factor 7A complex, and how viruses exploit this part of the pathway. And Henry and John, and not in that order, are going to tell you about how viruses participate in the coagulation pathway. So first up on the block, we're going to have Li Hua tell us about the differential role of a special part of clotting factor 10 and many other of the proteases and zymogens in the clotting pathway called the GLA domain. Take it away, Li Hua.
Thanks, Ed, for the wonderful introduction. Hello, everyone. My name is Li Hua Hao, and currently I'm a postdoc in Pryzdial lab. And today, my topic is differential rules of coagulation factor 10 glodomin during activation. So first, I will reintroduce the process of factor 10 activation. So actually, there are 3 tenases can activate factor 10. So for the initiation, we also call this extrinsic pathway. This is tissue factor 7A complex. For the amplification, we also call this intrinsic pathway. This is factor 8A, factor 9A complex. So these 2 tenases are physiological tenases, and both of them rely on the calcium and the phospholipid. And in addition to these 2, there's another one, the non-human tenase, named Russell's-Weber-Winham 10. We also call this RVV10. So RVV10 also can induce factor 10 activation at the presence of calcium, but independent of phospholipid. So once factor 10 being activated to 10A, so the 10A gonna help to generate the clot. So then I will give some background information about glodomin. So glodomin is one part of factor 10, so characterized by reach of gamma carboxyglutamate acid. So the known function of glutamine is to help binding factor 10 to the phospholipid at the presence of calcium. But the unknown function during the factor 10 activation still remains unclear. So here we proposed 3 possible function of glutamine during activation: the protein-protein interaction, the tenase recognition, and the conformational stabilization. To exclude the phospholipid binding function, so we designed all our experiment at the phospholipid-free condition, but with calcium. So to compare the function, to investigate the function of glutamine, so we have to prepare the normal factor 10 and glutamine-less factor 10. So there's a classic way. So the chymotrypsin, which is a serine proteinase, it can recognize the aromatic amino acid and remove the glutamine from factor 10. So because the factor 10 and glutamine-less factor 10 is already being used for the research for many years and commercially available, so we directly purchase this tool from the company. And then I conducted the 2-step chromogenic assay to check the factor 10 activation. So briefly, the factor 10 or glutamine-less factor 10 was added as substrate, and different tenase was added to initiate the reaction. And of course, this reaction is at the presence of calcium, but no phospholipid. So once the reaction started, so factor 10A or glutamine-less factor 10A will be generated. So I will stop the reaction at different time point and give the chromogenic substrate for factor 10A to check how much factor 10 was generated. So it means one-time experiment, I can get factor 10A production according to time. So this can give me the chance to calculate the reaction rate. We also call this velocity. So the velocity just corresponds to 1 point in this curve. So by repeating many times this experiment using different concentration for factor 10 or glutamine-less factor 10, so I can get many points according to the concentration of the substrate. Then I fit these velocities into the Michaelis-Menten kinetics analysis. Then I got the Michaelis-Menten curve, all shows in here. So here is a Michaelis-Menten result for 3 different tenase. The shape may seem quite similar, but if you focus on the X or Y axis number, you can find they are totally different. And I summarized all the analysis result on next slide. So before checking the final result, let's briefly review what is Michaelis-Menten curve and what can we get from this analysis. So Michaelis-Menten curve can reflect the relationship between the velocity and the substrate concentration. So we can get 2 key information. This blue line indicates the Vmax. It means the maximum reaction rate. So it means when we give enough substrate, when the tenase or enzyme is saturated, the maximum reaction rate means Vmax. So Vmax can reflect the catalytic capacity. So lower Vmax means lower capacity. And another information is Km. So Km means when the reaction rate reach half of the Vmax, the concentration of the substrate equals to Km. So Km usually reflects the substrate binding. So lower Km means higher affinity. So this is opposite. And then we can move to our result. So if we check Vmax, we found in all cases, the factor 10 has much higher number compared with glutamine-less factor 10. So this implies removal of glutamine gonna impair the factor 10 activation even at phospholipid-free condition. And if we check the Km, we found for 2 tenase, the tissue factor 7A and RAV10, the glutamine-less factor 10 has much higher Km compared with normal factor 10. So which indicates the weaker binding. But interestingly, for factor 8A and factor 9A, this complex, there's no significant difference between the KM. So this shows clotamine plays no rules for factor 8A, 9A complex. And finally, if we check the yellow part, we found the difference is not consistent. So we can get the conclusion the factor 10 is differentially recognized by each tenase. So to summarize my project, so first, the clotamine of factor 10 is critical for activation by each tenase, and independent of phospholipid, the clotamine is not important for the intrinsic tenase, which means factor 8A, factor 9A complex. And this provides opportunity for the therapeutic development targeting distinct factor 10 activation pathways. And in the future, I will measure the phospholipid independent binding of factor 10 to each tenase to clarify our KM result, and also determine the 3D structures for the tenase and factor 10 for binding intervention opportunities. So that's conclude all my presentation. And now I will hand it over to Alex.
Excellent. Hi, my name is Alexandra, and I'm going to be moving over to the fibrinolysis or clot breakdown part of this. Still, once again, focusing on factor 10. This time, as you can tell by the title, we're looking at a recombinant or lab-made form of factor 10, and I'm using it as a clot buster. So we'll start with a background on this fibrinolytic process, which is clot breakdown. And we're going to start by looking at the intact fibrin in purple there. You normally think of that as the meshwork of the clot, so that's going to block the flow of blood. And that will bring together our TPA, tissue plasminogen activator, and our plasminogen. TPA will cleave plasminogen into plasmin, and this is the real initiator of clot breakdown. So a little bit of plasmin, you'll start priming your fibrin, and with a lot more plasmin at the site of the clot, we're going to dissolve the clot. For this reason, when somebody has a heart attack or a stroke or otherwise a large clot, what we're going to be giving them as treatment is going to be this tissue plasminogen activator, TPA, sold under the drug name Alteplase. It's been used for about 4 decades now, and it's great. It works really well. As you can imagine, giving someone more TPA, you can generate more plasmin and break down the clot faster. However, you can already see on the right side of this box here, it has some problems as a medicine. We have not yet solved them. We're still using TPA, but unfortunately, we have to give our patients this huge bolus dose of TPA, which means that instead of locally acting just to break up that one big clot, you've got systemic effects, systemic bleeding, which can include cerebral hemorrhage, brain bleeds, and up to about 6 to 7% of patients. For that reason, we're looking for an alternative, or at least something to lower that dose. And that is where factor 10 is coming in. Our lab discovered quite a few years ago now that factor 10 is, of course, the center of the clotting universe and is an auxiliary cofactor to this process. So this is the same thing we just showed you, moved it down, made some room for factor 10. So factor 10's role comes in once you start generating plasmin. You can see once we've got a little bit here, plasmin will not only start priming this fibrin and breaking down the clot, but it'll cleave factor 10 into a factor 10 beta form, which is then able to act as a cofactor here, bringing together more TPA, more plasminogen to break down the clot. How can we make this a good therapeutic? Well, we didn't want to just give you factor 10 because, of course, we've just heard about how 10 becomes 10A in a variety of ways, especially if you've been bitten by a Russell's viper. But we generated 10AIK, which is a chemically modified form of 10A. So this drawing we've got here, again, we've got that circle with its little dimple, which means that it's an active enzyme. You can see our GLA domain down here. What we've done in red here is attach this PEG spacer, which will not only help us attach a little C-terminal lysine on the end, but it inhibits that active site that 10AIK has. So if we give you this drug, you're not forming any more clots. We're just going to break them down. And 10AIK works really well. This is our first data here on the bench. We're looking at a y-axis of clot amount. So what we do here is we form a clot and then we watch it break down. And it breaks down pretty slowly if you follow this line here, where we haven't added anything in here. But if you add various amounts of 10AIK, your clot breaks down faster. This allows us to move into our animal model. So this is a preclinical model of carotid thrombosis. So we open up and expose the carotid artery of a mouse and cause a clot. What we're going to do here is measure the the blood flow of a mouse through a Doppler ultrasound. It gets hooked under their artery, and it'll read, for instance, a nice high blood flow to start. And then when we cause a clot, that blood flow goes down to 0. Once we inject our drug, for instance, 10-AIK got injected here, we wait a little bit, and then we can see blood flow restored or reperfusion about 15 to 20 minutes later. We tested this with a bunch of different titrations. So we had just the TPA that you would normally give a mouse or a human, and then just the 10-AIK, and it was very successful. So that would be efficacy. In the safety model, again, preclinical trial, we're looking at biomarkers of systemic bleeding. So again, the main problem with TPA is that it causes systemic bleeding. So that is bad. And what we can see in these blots here, these are 6 individual Western blots with 5 lanes or 5 mice each, where we're starting looking at giving a mice no drugs, no TPA, no 10-AIK, and we're looking for lytic fibrinogen fragments. And we can see that there are none here. But if we give the mice a full dose, let's say 17 micrograms per gram of TPA, we are starting to see these lytic fibrinogen fragments. And this says to us, not a safe dose here, there is systemic bleeding. So as we reduce the amount of TPA or replace it altogether with 10-AIK, we don't see those fragments anymore. So this is a safer drug from that perspective. This is fibrinogen and this is plasminogen, and both of them depict safer qualities with that 10-AIK. Unfortunately, 10-AIK is quite difficult and complicated to produce as a pharmaceutical. The first is that we're taking factor 10 from plasma and then chemically modifying it. There's a lot of challenges there. And so what we want to do instead, and why I mentioned at the beginning a recombinant drug, is make a lab-made version of this. We want it to act the exact same way, but not be so chemically modified. So we're going to do some mutations instead. So that allows me to introduce what I call double mutant RFX-IC. So the little R stands for recombinant or lab-made, factor 10, and the little I and C are the 2 mutations that I've made. So the first one, the little I, is an inhibitory mutation. So again, factor 10 can become 10A. We don't want that. The first thing we look at is this little catalytic triad that our serine protease has. If we swap that serine out for an alanine, we've inhibited that clotting site. The next mutation, the little c, is a cleavage-resistant mutation. Here I'm depicting something similar to what I showed you before, where I said that plasmin will cleave factor 10 into 10 beta. This is really good news for us. It has a golden C-terminal lysine on the end. That same thing we added to 10-AIK. We think that's really important for the mechanism here. Plasmin will cleave 10 beta into 10 gamma. It does receive another C-terminal lysine. However, this form is rapidly cleared by the system, so we don't want that second cleavage. So our cleavage-resistant mutation takes a lysine to a glutamine to inhibit that. First thing to do when you make a new drug is see if you actually made what you think you made. So what I wanted to do here was check that my mutations were successful, the first one being that inhibitory mutation. So we can check that out using a factor 10A clotting assay or a prothrombin time assay like you might see in the clinics. What we have here is factor 10 deficient plasma. We can add in factor 10 of our choice and record our time to clot formation. So this is clotting time on the y-axis here. If you add no factor 10, you can't form a clot at all. We need that. If you add in wild type factor 10, plasma will clot in about 12 seconds. So this is our very well-established average in the lab. Then we can add in our double mutant or the single mutants. I, of course, had to make one of each. Again, we're looking at mechanism throughout this whole process. If you have a little I mutation, whether alone or in conjunction with the other, the factor 10 is not able to become 10A. It's not able to form a clot. So that's kind of a check mark on our list there. And I also did want to see if the little c on its own had any activity. And it still did. It didn't change anything there. So that's also pretty good. The next checklist item is the little c mutation. So does the cleavage process still happen? These, again, are some blots. We're going to look at the left first at the wild type factor 10. We can see there's factor 10, 10 beta, and then factor 10 gamma over a period of plasmid incubation. The little I mutation does not change that. We see all 3 forms. And then another check mark, anything with the little c, whether alone or in the double mutant, we get the factor 10 beta that we do want, but we don't see the factor 10 gamma. Now I get to test it in the lab. First we do in vitro. We're going to be looking at how it works. So again, we think that as plasmin gets generated, it generates factor 10 beta, and then that's going to generate even more plasmin. So now we're going to look on the y-axis at plasmin generation over time. And black at the bottom here, if we add nothing, just that TPA and the plasminogen, how much plasmin can it generate? If we add any of our factor 10s, we're generating much more plasmid, so it is accelerating that process. We can then look at a clot lysis assay. So this is the same thing, but a little bit more colorful that I showed you for 10-AIK. So this is clot strength. We watch the clot formation and then clot breakdown. Again, it's pretty slow up here if you add no factor 10 or just the wild type or single mutants. But as soon as you add in blue here, that double mutant factor 10, we're starting to see clot breakdown happen a lot faster. This allows me to move into our preclinical mouse models. Again, the exact same model with the carotid artery. Now we're looking at time to complete reperfusion. So when does the blood flow return? At first, we have no drugs, so the blood flow does not return. Everything is completely occluded at 45 minutes. And then we're going to be titrating down. So we start with the gold standard, 20 micrograms per gram of that TPA. It will break down a clot and restore blood flow in about 15 to 16 minutes. If you go to a subtherapeutic dose, you cut that in half, no activity, we cannot bust that clot. If you just use our factor 10 on its own, you also can't break down a clot. But here, if we titrate the two of them together, so for instance, a subtherapeutic dose of TPA with a little bit of our factor 10, we're going right down to that 15 to 16 minute reperfusion time. And then we can kind of titrate that up as we go. So for this section, what we're looking at is clot breakdown, clot busting. We started with 10-AIK to try to replace TPA or at least reduce it. Mild success there. On the other hand, we could not continue with the project, so we made a recombinant version, the double mutant here. So far, it looks like we made exactly what we think we made, and its efficacy is good, but our next step is safety. So actually this week and the next coming weeks, we're looking at doing those Western blots again, looking for those systemic biomarkers. And then we're going to move into some more biochemical characterization and see what else happens after that. So now we'll move into the virus side of the lab, and I'll invite Henry up.
All right, thank you, Alex. Like she mentioned, I will now be talking a little bit about the virus side of the lab and how we are investigating viruses' use of tissue factor. So envelope virus infections commonly induce defects in hemostasis, and a few of these viruses and their effects are fairly well known. So if you think about SARS-CoV-2 and 1, which are prothrombotic, or hemorrhagic fevers that include Ebola virus and dengue virus. But it isn't just these viruses. Almost every single envelope virus has been documented to affect hemostasis. And we wanted to propose possible mechanisms for these observations. So first, a quick explanation about what an envelope virus is. Envelope viruses are a type of virus that incorporate a lipid bilayer in its structure, which is derived from the infected host cell. And the source of this envelope depends on the virus that is infecting the cell and can be derived from the endoplasmic reticulum or the Golgi or the plasma membrane, among others. And alongside the virus-encoded proteins that are embedded in this envelope, it can also incorporate host-derived proteins, which were embedded in the membrane before the virus budded off of the cell. One of these proteins is tissue factor, which we've already heard a little bit about. And briefly, it functions as a cofactor for the factor 7A mediated activation of factor 10 to factor 10A. And downstream, this results in the activation of thrombin and the formation of a clot. Tissue factor, though, is also involved in cell signaling through activation of the protease-activated receptors. And this occurs through either the proteases in the tissue factor complex or that thrombin that I just mentioned was generated downstream. And these play a role in a number of different processes, both disease and physiological. Now, released from its normally tight regulation, tissue factor on the surface of viruses can also induce these processes, both clotting and PAR activation. But how many viruses actually have tissue factor on their surface? Tissue factor, whether its expression is constitutive or induced, is expressed on a large variety of cells throughout the body. And this is certainly not an exhaustive list down here, but many envelope viruses do infect these tissue factor producing cells and thus likely incorporate tissue factor in their envelope. So how do we confirm that tissue factor is on the surface of these viruses? So we've been employing immunogold electron microscopy for this purpose, which allows us to visualize antigens on the surface of the virus by conjugating an antibody against these antigens to a gold bead, which appears like a little black dot on electron microscopy micrographs. These antigens that we're interested in, of course, include tissue factor, as well as viral proteins that mark that particle as a virus. And we also investigated the presence of anionic phospholipid through annexin-5 binding. And these pictures are showing HSV1 herpes virus, which is staining positive for tissue factor, as well as the viral protein, glycoprotein C, and anionic phospholipid. And we can see here that this variant is staining positive for all three of them. And the lab has also observed tissue factor on the surface of other herpes viruses, including cytomegalovirus and HSV2. So there is tissue factor there, and it may contribute to the activation of coagulation. But what else can it do? Like I just said, it can activate those protease-activated receptors. And this can result in an increase in viral infectivity. So these cells were treated with either thrombin or a vehicle, and then infected with HSV1. And there's a significantly increased amount of viral plaques that we can observe in the thrombin-treated cells. And this trend is similar when these cells are treated with factor 10A or factor 7A as well, which are other members of the tissue factor complex. Now, do these results hold in vivo? Well, in a murine model of HSV1 infection, administration of anticoagulants, which target either thrombin or various other members of the tissue factor complex, significantly reduced the viral loads in every single organ that was tested. Further investigations with tissue factor negative virus, which was grown in a tissue factor negative cell line, showed that the virus lacking tissue factor infected poorly compared to tissue factor positive virus. And this suggests that tissue factor may be a viable target for the generation of broad spectrum antivirals, or even the repurposing of current therapeutics for this purpose. So we aim to increase the number of viruses known to contain tissue factor in their envelope by examining these three viruses. They're from three diverse virus families and include dengue virus, HIV, and the common cold virus, HCOV229E. And they all cause significant burdens to the global healthcare systems and result in large amounts of economic damage. Dengue virus is a hemorrhagic fever and can cause an inability to maintain hemostasis, while HIV infection is associated with a significant increase in thrombosis incidence. Now, we grew up and purified batches of these viruses to begin the in vitro investigations. The first step was to examine the preparations of the virus and determine whether they are clean. These are some representative images of an HCOV229E and dengue virus prep, and they both appear to have viruses that are the right size and morphology. And they are both clean and free of any cell debris. They just contain viruses. The next step is to perform immunogold electron microscopy on these preparations to determine if the viral envelopes contain tissue factor. As we can see here, dengue virus co-stains for both a viral envelope protein, E protein, and tissue factor, and also stains for anionic phospholipid. Both HCOV229E and HIV both stain positive for tissue factor, though we're still working on these viruses. And this isn't just an artifact that can be observed in lab-grown and purified viruses. We've also observed tissue factor on the surface of viruses straight from infected patient plasma without any manipulation by us. HIV co-stained for the viral glycoprotein 120 and tissue factor, while hepatitis C virus stained positively for E protein as well as tissue factor. So this isn't just something that occurs in the lab as a result of growing up in tissue factor positive cell lines, but also something that actually occurs during viral infection. So tissue factor is present on the surface of the envelope in a variety of different envelope virus families in both lab and patient-derived samples. Now, if tissue factor inhibition reduces infectivity among other virus families, it could represent a broad spectrum antiviral target. Now, to that end, I am currently generating a humanized model of dengue virus infection in mice, which I'll use to assess the effect of tissue factor pathway inhibition on viral infectivity and thrombocytopenia, which is an important clinical component of dengue virus infection. And now I'll pass it over to John, who's going to tell you a little bit more about tissue factor activity on the surface of these viruses.
Awesome. Thank you, Henry. So building off of the work of identifying tissue factor antigen on those 3 virus types, so members of the Coronaviridae, Flaviviridae, and Retroviridae family. Next, we wanted to look at whether or not this tissue factor was functional. So the first step was to look at whether or not it retained cofactor activity. So this was done in a factor 10A generating chromogenic assay. And in here, we add purified virus as our source of tissue factor, incubate it with a purified factor 7A, factor 10, and calcium, and allow for the generation of factor 10A, which can cleave a chromogenic substrate and produce a color change that we can measure. So across all 3 virus families, we can see that as we add more and more virus particles, there's an increase in 10A production. So all 3 of these virus families are capable of accelerating factor 7A and leading to the production of 10A. So in order to implicate tissue factor from the virus in this 10A production, we did this using specific inhibitors, so a monoclonal antibody to tissue factor 9B4, as well as a protein from a bloodworm, NAPC2, which inhibits the tissue factor 7A complex. So when the virus is added alone and incubated with 7A, factor 10, and calcium, we can see that there's 10A being produced. However, when we take away virus, there's no 10A being produced. When we incubate it with an anti-tissue factor antibody, this significantly reduces the 10A. And that same effect is not observed when we add an isotype IgG control. However, NAPC2 is also able to significantly reduce the 10A being produced. And we see this again in all 3 virus families. So following this, we know that these viruses are capable of generating that procoagulant factor 10A species. We want to see if downstream this 10A can participate in thrombin generation and subsequently fibrin clot formation. So in order to do this, first, before moving on to the clotting assays, we've also implicated anionic phospholipid, another component of the extrinsic 10A complex, by using lactoferrin. So lactoferrin is a glycoprotein which can bind to anionic phospholipids and act as a competitive inhibitor for the binding of 7A or factor 10. So in this 10A generating chromogenic assay, we can see that virus on its own is able to produce 10A. However, when we add lactoferrin, this brings it to the baseline and there's less 10A being produced. So moving on into clotting assays in order to implicate tissue factor in the generation of thrombin and fibrin clot formation, we used pooled plasma and added virus as our source of tissue factor and initiated these reactions with calcium. And we can see that the more and more virus you add, the shorter the clotting time is resultant. So there's a dose-dependent reduction in the clotting time, and this is observed in all 3 virus types, so dengue, human coronavirus 290, as well as HIV. And to implicate tissue factor in this clotting activity, we again incubated with a monoclonal antibody to tissue factor 9B4, as well as NAPC2. And for dengue, we can see that the clotting time there's no clot formed when we take away the virus. When the virus is present and it's incubated with an anti-tissue factor antibody, there's also no clot formed past our cutoff of the assay. But this result is not observed with a non-specific IgG isotype. And the same is observed for NAPC2. And we see these results being accompanied in the other 2 virus families as well. So it's a common mechanism that's shown on these 3 very distinct families of viruses that are implicating tissue factor as a good target for broad spectrum antiviral therapeutics. So the take-home messages are that viral tissue factor is sufficient for clot formation in plasma, that there's also host-derived procoagulant anionic phospholipid on these viral envelopes, and that the tissue factor activity is present in these 3 distinct families. So things I'm currently working on are using CRISPR to generate a tissue factor deficient cell line that I'm able to produce dengue virus in and purify it. And I'll be studying the pathology and infection of dengue and HIV using this cell line. And now I'd like to bring Ed back up to cover today's punchlines.
Weren't they all great? I think they were fabulous. I'm lucky. Today's punchlines. We had Li Hua tell us that there's differences in how factor 10 is recognized by the tenaces in mammals, giving us therapeutic opportunities to differentially turn one or the other one off. We had Alex being able to tell us that we've got a novel opportunity to develop a new thrombolytic therapeutic, reducing the risk for cerebral hemorrhage in up to 6% of patients who need these drugs. And we had Henry and John tell us that we may have a broad spectrum opportunity for an antiviral agent. Now, wouldn't that have been nice 5 years ago when we had no opportunities when this nasty thing called SARS-CoV-2 hit us? This is the rest of our team. A number of these people have already left us, but I leave them in the family in this photograph because we continue to collaborate for a time being. And we have some incredible collaborators who are facilitating these projects, both here at UBC as well as outside of University of British Columbia. I put this picture in all the time to remind us how lucky we are to be where we are. I remind you to follow that path when you find it. You just never know what you're going to find. Keep your eyes open. I took this from my deck when the snowbirds flew by. Keep your eyes open also for nasty things. Anybody know what this is? That's poison ivy. It's all over the place in some parts of BC here. Can you see this gopher snake? Keep your eyes open. We're happy to answer questions. Please.
From the first presenter, so can I ask why you measured the Vmax instead of Kcat or Kcat-Km? Because Vmax depends on the enzyme concentration. If you a little bit changing enzyme concentration, the Vmax will be like up and down. So, but Kcat has this advantage.
Would you like me to handle that? The enzyme concentration is the same in each of those. So that means we're essentially comparing Kcat.
Okay, thanks.
That was easy. Thanks for letting me answer.
Just out of curiosity, because I'm ignorant to the topic, the tissue factor that you're finding on the surface of the different of the different viruses, what's your ideal strategy for leveraging that to produce new antiviral agents?
Either of you guys want to handle it? Hell no. Tissue factor is a tricky cofactor to try to target specifically. If you turn it off completely, you risk bleeding, just like any other anticoagulant. Tissue factor is the initiator of coagulation, so you especially risk the possibility of bleeding. The option that we are interested in is something that we didn't talk about at all. And that is that viruses that we have investigated also express cofactors for tissue factor. And we will be in the future investigating the idea that inhibiting the helpers of tissue factor on viruses is going to be the ticket. So we'll be able to very specifically turn the mechanisms on the virus only off. Tell me that's not arm waving, and you'll owe me something. Nevertheless, we have the very first common antigen on all of these viruses, and that's something to work with.
Thanks so much for the talks, guys. I was kind of just wondering from John's data set, it seemed like some of the patterns in the HIV versus the dengue virus and the human coronaviruses were a little bit different. Yeah, and if there are any thoughts or hypotheses there.
So one runner compared to the other runner, you better answer this question.
Thanks for the question, Matt. Yeah, so are you referring for the 10A generation? Like how for HIV, that 10A generation was done in the linear range of HIV. So we've just done that on a small batch of HIV that's produced. So we will be in the future on a new preparation, be having a more similar curve to what we see in dengue and human coronavirus 210E. And then the same for the clotting, the reduction in clotting time. It's just 3 points there.
I guess also the anti-tissue factor antibody in the last
Yeah. So it's just been done with NAPC2, and that's again because of limited HIV samples. So from that preparation, so those are just more preliminary data with just NAPC2 as the inhibitor, but we would have to do that with anti-tissue factor and do a titration to figure out the optimal concentration for inhibition.